So, in today's lecture, we will look at the various antiviral agents. So, to begin with, viruses are actually very interesting organisms. They are actually non-cellular infectious agents, and they need a host cell for them to survive and multiply. So, there are of course, wide variety of viruses which are capable of infecting bacterial, plant or animal cells, and there are about 400 that are known to infect humans. So, the ones that are infecting humans or animals are called as zoonoses, and they can be transmitted in many ways. So, the most common viral infection that many of us would have encountered is the flu, which is basically the common cold, which is influenza, and there are also other viruses which we may have encountered in one of our family members, which is chickenpox. Measles, mumps are also not very uncommon, and you have viral pneumonia, you have another disease called as rubella, and of course, the major disease that we encountered in the past, which is smallpox. And all of these can be transmitted through the air, so by sneezing or coughing. So, if one gets in close proximity to an infected person, then there is a good chance that we will acquire this infection through or acquire this virus through sneezing or coughing. So, which is why these diseases are actually quite contagious. There are other kinds of viruses which can be transmitted by the means of a carrier, just like you have malaria, which can be transmitted through mosquitoes. There are also others viral infections which can be transmitted through arthropods or ticks. Okay, so, the example is this Colorado tick fever and yellow fever, which is actually transmitted by these organisms. Some viruses are unable to survive for long outside the host, okay, and are transmitted through physical contact. So, for example, the major epidemic in the world, which is AIDS, which has affected millions of people in the past 30 years, is actually transmitted through physical contact or through blood transfusion. And the common cold has also gone through physical contact. Genital herpes, certain kinds of leukemias, and rabies are examples of transmitting the virus through physical contact. Finally, they can also be food or waterborne diseases, which are such as hepatitis A and E, poliomyelitis, which is again it's been eradicated in India through vaccination, but it is a waterborne infection. And you can also have viral gastroenteritis, which is quite common occurrence in during floods and heavy rains. Okay. Historically, viral infections have proved devastating, and it has led to the death of millions of people. So, the smallpox, for example, has been suggested to be one of the reasons why the Roman Empire, which was very strong in the first century, and it started declining because of smallpox. Smallpox was also responsible for the death and destruction of a number of indigenous tribes during European colonization. So, what happened was especially in South America, smallpox was not prevalent, and when the colonization by Europeans occurred, there was a lot of death and destruction associated because this virus was transmitted to the population. In some areas, it is estimated that 90% of the population died from this disease. Another major historical event which happened in the 20th century is the flu pandemic, in which about 20 million people died. Okay. This happened in the years 1918 to 1919, and it is still fresh in the memory of our medical community. And this number, which is 20 million, is far more than what was than the number of people who were killed during World War I. Okay, so just to think about this, put this in perspective, this kind of a disease can cause death and destruction to millions of people, and which is much more than what would happen if there is a military conflict. So, therefore, there is a possibility that a large population of people in a country can be wiped out if the infection is quite severe. So, HIV infections, for example, which leads to AIDS, has been around for about 30 or 40 years, and it's resulted in the death of more than 30 million people. The Ebola virus, for example, is another major problem. especially in Africa. And the spread of viral infections is nowadays quite large because of globalization, and there is a large number of people who are sort of traveling even on a daily basis across the world. And so, if there is a section of population which is affected by a virus today, they can quickly spread to another far away region because of the rapid movement of people across the world. So, in recent memory, the outbreak of severe acute respiratory syndrome, which is SARS, is something that we would want to discuss. So, this happened in the Far East in the year pretty much in the year 2003, and there was immediate action taken by the medical community, and that resulted in a lesser of an effect. But if it had been ignored, then it is quite possible that a much larger number of people would have been affected by this disease. So, this brings us to the possibility of bioterrorism. So, here what can happen is that there could be terrorists who will introduce the disease into a population with the intent to kill the population or get rid of the population with viruses. So, which is why we need to keep our guard up, and we need to be able to develop effective antivirals, even vaccines for that matter, so that we can counter the possible damage that these kinds of viruses can cause. So, let us start with general understanding of structure of viruses, and viruses are basically protein packages, and these can transmit foreign nucleic acid between host cells. Okay. So, this is how we can very simplistically look at it. There are mainly two kinds of viruses. One is the ribonucleic acid or RNA virus, and the other one is the DNA virus. So, but a virus will not contain both, that is, it will either contain RNA or DNA. So, here is a structure of a virus, general structure of a virus, and most RNA viruses contain a single strand of RNA, okay, and most DNA viruses contain double strand DNA. So, that is pretty much like what is there in our mammalian cells. The size of the nucleic acid varies widely, with the smallest viral genome coding only for 3 to 4 proteins. Okay. So, imagine an organism such as a virus, where you have the genetic code coding only for 3 to 4 proteins, and they are sufficient to go into a host cell, infect a host cell, and take over the host cell machinery and lead to potentially fatal consequences. And the largest coding virus is over 100 proteins. So, these are far lower than what a single cellular organism that we are familiar with can actually code for. So, viruses are extremely simple molecules or simple organisms, but they can have devastating consequences. The viral nucleic acid is contained and protected within a protein code called as the capsid. Okay. Capsids are usually made of protein subunits called as protomers, which are generated in the host cell and can interact spontaneously to form a capsid in a process called as self assembly. So, here is the capsid, and these are ones that are going to coat the nucleic acids. So, once the capsid that contains the viral nucleic acid, then the whole assembly is known as a nucleocapsid. So, this is called the nucleocapsid, and in some viruses the nucleocapsid may contain viral enzymes which are critical to its replication in the host cell. Okay. The flu virus, for example, contains an enzyme called as RNA dependent RNA polymerase within its nucleocapsid. There could be additional membranous layers of carbohydrates and lipids surrounding the nucleocapsid, which is shown here, and basically there would be variation depending on the virus concerned. Some of these are viral proteins which have been coded by viral genes, but most are host derived proteins. So, this is a general structure of a virus. The complete structure is known as a virion, and this is the form that the virus takes when it is outside the host cell. So, the size of these virions is between 10 and 400 nanometers, and so if you want to look at it under a light microscope, it is not possible. So, you would need an electron microscope to detect the presence of viruses. Now, let us look at the various stages in which the infection occurs. The first step is known as adsorption. So, adsorption is when the virion is going to go and bind to the outer surface of the host cell. So, this involves a very specific interaction and specific molecules on the outer surface of the virion that helps with binding to the specific protein or carbohydrate that is present on the host cell membrane. Okay. So, this host cell protein or carbohydrate can be called as a receptor for the virion, and the host cell has not produced this molecule to be a viral receptor, of course, but it can serve as a way in which the virus is going to interact with the host cell and start getting adsorbed. And these molecules which we already know are called as glycoproteins, and they have crucial cellular functions such as binding of hormones. So, this is a place where some of the hormones are going to interact with the cell surface and help with signal transmission. So, the virion takes advantage of these, and once it is bound, the next stage can take place, which is the introduction of the viral nucleic acid into the host cell. So, the next stage is penetration and fusion. So, different viruses introduce the nucleic acid by different methods. Some of them inject the nucleic acid through the cell membrane. Okay. Others enter the cell intact and then they are uncoated. So, depending on what the type of virus is, this mechanism will change. So, the viral envelope of some virions fuses with the plasma membrane, and the nucleocapsid is then introduced into the cell. So, this is the process of fusion, and then once it gets in, it is uncoated and goes to the endosome. So, after the endocytosis takes place, then the cell membrane wraps itself around the virion and then is pinched off to produce a vesicle called as the endosome. Then fusion of this endosome with lysosome and host cell enzymes aid the virus in the uncoating process. And since we have discussed previously, the endosome has a lower pH and it also triggers the uncoating. Then comes the major process of replication and transcription. So, the viral genes that can be defined as in two stages. One is the early stage and the second one is the late stage. So, the early genes take over the host cell such that the viral DNA or RNA is synthesized. Okay. So, here is the process in which the virus is going to use its machinery, incorporate itself into the host machinery and replicate, okay, and divide and produce a larger number of viral particles. So, the viral enzyme, which is called as RNA dependent RNA polymerase or transcriptase, is now going to help with synthesizing the mRNA, okay, and these mRNA code for viral proteins. So, this is the key step that we are discussing right now, which is replication and transcription. Then what happens is that you have, after these proteins are synthesized, then you have the synthesis and assembly of nucleocapsids. So, we already discussed that nucleocapsids are the outer membrane which is important for holding the nucleic acids, and so these are also synthesized, and these are done by the so called late genes. Okay. So, the late genes direct the synthesis of this capsid proteins, and then there is a process of self assembly which forms the capsid. The viral nucleic acid is then taken into the capsid, which is shown here, and they form a nucleocapsid. Then comes the next process, which is basically the virion release. Okay. So, the naked virions, that is with no outer layer around the nucleocapsid, are then released by cell lysis, and then the cell is actually destroyed in this process. And this can be done by a process known as budding. Okay. So, here is the budding process, which is then going to result in the release of the virion. So, just to recap, the first step here is adsorption, wherein some of the cell surface receptors are recognized by the virion, and the virion goes and binds to this. Okay. Once it is binding, then it forms, it penetrates and fuses. It is also possible that the nucleic acid is injected into the cell, but in this example that we are looking at, it is the entry of the entire virus. So, once it gets in, then there is an uncoating that happens, and then it incorporates into the host nucleic acid machinery, and then there is replication and transcription. Now, this replication and transcription results in the generation of multiple copies of the virion, and this is now it is coated by a nucleocapsid, and then by the process of budding, it actually is released. And in this process, the host cell is lysed, and therefore, the host is dead. Okay. So, one of the ways in which we can protect ourselves from viral diseases is to use vaccination. And number of diseases that we looked at such as polio, measles and mumps are actually mainly targeted by vaccination. So, smallpox and yellow fever are also sort of serious diseases which are countered by vaccination. So, vaccination works by introducing the body to a foreign material, okay, and this foreign material has very similar molecular structure or it has a component of the virus. Okay. So, what happens is that our immune system generates or recognizes these as foreign and starts generating antibodies for eliminating this new material. So, therefore, we provide the opportunity for the molecular fingerprint of the virus to be able to encounter the immune system, and now the immune system is now primed to attack the virus and which can infect the body. Okay. So, this is one of the ways in which we can effectively counter viruses. So, this vaccine can also contain a killed or weakened version of the virus. So, therefore, this itself will not lead to infection, but it can introduce into the body a structure which is now going to be recognized by our immune system to produce the corresponding antibody. There could also be fragments of the virus which are known as subunit vaccines which can be used. And typically, this is a highly preventive approach, and once the person is actually infected with the virus, one cannot vaccinate this individual. Now, if the person is affected by a viral infection, then since we cannot vaccinate at that stage, we need to start administering antiviral drugs. So, now we will look at how these drugs have been developed. So, there are a few diseases where there is no effective vaccine, and so therefore, we need to use a drug. So, for the most time, the virus is effectively disguised from the immune system and from circulating drugs. So, since it uses the host machinery, designing the new drugs is actually quite challenging, because we do not want to kill the host as well as the virus, but we want to only kill the virus. So, again, as we looked at previously, there are two kinds of viruses. One is the DNA virus and the other one is the RNA virus. And so, now we will look at some of the antiviral drugs which are used against DNA viruses. So, most of the drugs which are active against DNA viruses have been developed against herpes viruses, and these combat diseases such as cold sores or genital herpes, chicken pox, shingles, eye diseases, mononucleosis, Burkitt's lymphoma and Kaposi's sarcoma. So, here nucleoside analogues have been particularly effective. So, the first class of drugs that we are going to encounter are inhibitors of viral DNA polymerase. So, the first drug that we are going to look at is acyclovir. So, acyclovir triphosphate, what we have already looked at this previously, is an incomplete nucleic acid, and therefore, it is going to prevent DNA replication. So, the way in which this happens is that there is this is the prodrug, it gets phosphorylated, and then it encounters the DNA growing double strand, and then during replication, and then it prevents the replication from occurring. Okay. So, this is the classic example of a chain terminator. Now, the question is that we would need to address is, how is this molecule going to be selective? That is, how is it not going to affect the DNA polymerase in normal uninfected cells? So, the first phosphorylation reaction that occurs is by this enzyme known as thymidine kinase, and here this enzyme is present in host cells as well as the herpes virus. But it turns out that the selectivity towards the herpes virus, the viral thymidine kinase, is about 100-fold higher. So, therefore, the thymidine kinase which is present in viruses is actually more effective in doing this phosphorylation. So, compared to the host cell, and therefore, this is one reason why it is selective. Also, there is a selective uptake of acyclovir in infected cells, and together this explains the excellent activity and reduced toxicity relative to the previous drugs that were developed in this area. Another feature is that it shows a 50-fold selective action against viral DNA polymerase relative to cellular polymerases. So, therefore, this drug is one of the most important drugs that have been developed against herpes viruses. One of the problems with acyclovir is that it has very low oral bioavailability. It's only about 15 to 30%. Okay. So, again just to recall, oral bioavailability is basically after one consumes the drug, how much of the drug is available in the bloodstream. So, therefore, about 85 to 70% of the drug is not available, right. So, in order to address this, another analog, which is the L-valine ester of the molecule, was actually generated. So, this molecule has better absorption from the gut compared to acyclovir. However, the polarity of this molecule is similar to that of the parent drug. Now, when we use the D-valine derivative, then what happened is we observe diminished absorption. Okay. So, this observation suggests that there must be a specific binding interaction that facilitates absorption across the gut. Okay. So, now together what this means is that it is possible that this drug accesses certain transporters, because the L-valine molecule is recognized better by the transporter, it is pulled in. When the D-valine derivative is used, there is diminished absorption, and this is possibly due to poor binding of the molecule to the transporter. The transport proteins that are present in the gut are responsible for transporting dipeptides across the cell wall. Okay, and these for example, this HPEPT1 and HPT1, these are dipeptide and tripeptide transporters, which are possibly the ones which are taking this drug in. The next molecule in this series is desciclovir, and this lacks the carbonyl group that is present here. So, if you look at this structure, it does not have the carbonyl group. So, once it enters the blood supply, there is an enzyme which is known as xanthine oxidase, which oxidizes this position to give you the carbonyl derivative. So, therefore, this molecule is actually a prodrug. Okay. The next class of molecules is the ganciclovir, which is again an analog of acyclovir, and it has an extra hydroxymethylene group as shown here. Okay. So, and there is another version of this, which is valganciclovir, which acts as a prodrug for this molecule. It has the valine ester of this in the structure. So, there is another molecule which is again a prodrug, which has a diacetate derivative, and so this diacetate is cleaved by esterases to produce the free diol, which is then subsequently inhibiting the transcription. So, again the thymidine viral thymidine kinase is going to phosphorylate here and then subsequently generate the triphosphate. So, the two alcohol groups they are masked as esters, and this makes the structure less polar, and therefore, it is better absorbed. So, once it is absorbed, there is an enzyme called as aldehyde oxygenase, which then oxidizes this to give you the final molecule, which has the carbonyl group. One can also think about inhibiting tubulin polymerization. We have looked at this previously, and this can be one method by which you can inhibit viruses. And this is the molecule that we are looking at, which is podophylltoxin, and this has been used clinically to treat genital warts caused by the DNA virus. There are antiviral drugs which act against RNA viruses, which is the second class of viruses, and here we will specifically look at HIV. So, HIV is an example of viruses, of a group of viruses known as retroviruses. Okay. And there are two major variants. One is the HIV-1, which is responsible for AIDS in much of America, Europe and Asia, and there is another one which is HIV-2, which mainly occurs in Western Africa. So, a number of antiviral drugs have been developed in the past 30 years or so, and these help in slowing down the disease, okay, but they do not completely eradicate it. Recently, it has been shown that 2 people have been completely cured of HIV, and these are very promising. This looks very promising, and therefore, it is possible that in future we may be able to completely eradicate this disease. Okay. So, most clinically useful drugs target the viral enzymes known as reverse transcriptase and protease. So, here we need to develop effective drugs against a third target, and for this we need to have a good knowledge of the life cycle of HIV. Okay. So, let us look at this briefly. HIV is a RNA virus which contains a single strand of RNA, and there are two identical strands of this single strand RNA. And the viral enzymes reverse transcriptase and integrase, as well as other proteins which are called as P6 and P7. Okay. So, these are all enzymes which are present in this virus. The capsid is made up of protein units known as P24, and surrounding the capsid there is a layer of matrix protein known as P17, and then a membranous envelope which originates from the host cells, okay, and which contains the viral glycoprotein GP120 and GP41. Both of these proteins are crucial to the process of adsorption and penetration. Now, what happens is that GP41 traverses the envelope and is bound non-covalently to GP120, and then interaction with the host cell surface and subsequent conformational changes result in the fusion of the viral particle. Okay. So, then once fusion has taken place, it releases the single strand RNA. Before that, you need the enzyme called as protease, which breaks down the protein capsid, and then once it is broken, then you need to have reverse transcriptase. Okay. So, the released viral RNA is not capable of coding directly for viral proteins or for self replication. Instead, it is converted to DNA using the host machinery. So, viral RNA and enzymes are then released into the cell cytoplasm, and then it is converted to DNA and incorporated into the host cell DNA. So, the conversion of RNA into DNA is not a process that occurs in human cells, and therefore, there are no host enzymes to catalyze this process. HIV carries its own enzyme known as reverse transcriptase in order to carry this out. Reverse transcriptase is a member of a family of enzymes known as DNA polymerases, but it is unusual that it can use RNA strand as a template. The enzyme first catalyzes the synthesis of a DNA strand using viral RNA as a template, and this results in a DNA RNA hybrid. Then reverse transcriptase degrades the viral RNA, resulting in a single stranded DNA. Now, this single stranded DNA is used as a template to generate a double stranded DNA, which is also known as proviral DNA. Proviral DNA is then spliced into the host cell's DNA by an enzyme known as viral protein integrase. Once the proviral DNA has been incorporated into the host cell DNA, it is called the provirus, and this can remain dormant for weeks or even months or years together, okay, until there are certain cellular processes which are activating it. So, once this activation happens, then it results in transcription to produce viral RNA, which is shown here, and this viral RNA, some of which will be incorporated into new virions, and the rest of which will be used in translation to produce 3 large non-functional polyproteins. Okay. And then the first of these polyproteins is cleaved by cellular proteinases and produces the viral glycoproteins, which are incorporated into the cell membrane. After a series of steps which we will not go into details about, the budding occurs and then the virus is released. Until 1987, there was no drug available against HIV. Most drugs have been developed to act against the viral enzymes reverse transcriptase and protease. However, a serious problem with the treatment of HIV is that the virus undergoes mutation extremely easily. Okay? And so, once a drug has been introduced, there is going to be rapid resistance developed to the antiviral drug. So, therefore, treatment with a single drug can have short term benefits. However, after the drug has been introduced and the benefits are seen, since the virus can rapidly mutate and develop resistance, it is preferred that in the long term a combination of drugs is actually used. So, today HIV therapy consists of a combination of drugs which are given for pretty much for the lifetime of the individual. So, the demands of any new HIV drug are quite high. One, it has to be taken over long periods of time. So, it has to have maybe years or decades even for that matter. This drug that is developed must have high affinity for its target, which is in the picomolar range. it must be effective in preventing the virus multiplying and spreading. This new drug should also show low activity against similar host targets in the cell, and it should be safe and well tolerated. And it must be effective against a large variety of viral isolates as possible, or else it only serves to select resistant variants. Then it also needs to be synergistic with other drugs, because we are going to give a combination of these drugs, it should be synergistic with them and compatible with the other drugs used to treat these opportunistic diseases and infections arising from the weakened immune response. The drug must stay above the therapeutic levels within the infected cell and in circulation, and it also must be capable of being taken orally with a minimum frequency of doses. Okay. And it should preferably be able to cross the blood brain barrier in case the virus lurks in the brain. And lastly, it must be inexpensive. So, developing a new drug for HIV is actually quite challenging. So, with that, let us look at inhibitors of viral reverse transcriptase. So, as the enzyme reverse transcriptase is unique to HIV, it serves as a very good drug target. However, it still remains a DNA polymerase, and therefore, one has to be careful about how selective the drug that we are developing is towards reverse transcriptase rather than our host cell DNA polymerase. Okay. So, various nucleoside like structures have been useful as antiviral agents. So, here are some examples. So, didenosine, and then this is a deoxyadenosine triphosphate, and then this is AZT, which is one of the first drugs that was introduced. It contains an azide ring here, or you can have a chain terminating group such as a sulfur here, and so on. Again, these are the broad class mechanism of action of these drugs we have already looked at previously, and they are inhibitors of reverse transcriptase. And there are also non-nucleoside reverse transcriptase inhibitors, which are known as NNRTIs. And what these do is that these are generally quite hydrophobic molecules, and they go and bind to an allosteric binding site in the enzyme. So, nevirapine is an example of this, and since the allosteric binding site is separate from the substrate binding site, these are non-competitive reversible inhibitors.